for the day 3 program for metabolomics today. So today, as what we did, the morning time, there are 4 presentations from Nathalie about the general concept and the introduction about metabolomics. And then I will give a kind of introduction presentation about the mass spectrometry based metabolomics. And then Robert Young, he is an NMR expert at EMSL, and then he will give a talk about the NMR-based metabolomics. And then William Q, he will be presenting about the mass data processing or metabolomics data processing before working on the data interpretation or biological understanding. And then in the afternoon, we will have 3 subtopics. One is GCMS data processing, and the next LCMS, and then NMR data processing. Some softwares are not freely available. There's not a version we can use. So that's why NMR side, we will be showing the slides and also online streaming about some process. So before going to the Nathalie's introduction of metabolomics, Leon or Scott, do you have any high-level announcement or updates for the day 3?
I do not. Just keep putting your questions in the Discord or chat, I think, and although Discord's, I think, a little easier and persistent, and just enjoy yourselves and enjoy the talks.
Okay, sounds great. Then Nathalie, now it's your turn.
Hi, good morning. I am Nathalie Munoz. I am a scientist at PNNL, and I will be talking about an introduction to metabolomics today. So to start, what is metabolomics? So metabolomics is an omics science that enables the characterization of metabolites from cells, organs, tissues, or biofluids. It does this by using advanced analytical chemistry techniques. The metabolome, which is what metabolomics studies, is in turn the complete collection of metabolites or small molecule chemicals, which have molecular mass that is smaller than 1,500 Daltons, that are found in given organelles, cells, organs, biofluids, organisms. The term metabolome was coined in 1998 by analogy to the genome, transcriptome, and proteome. And these metabolites or small molecules are chemically transformed during the metabolism. In contrast to genes, which functions are subject to epigenetic regulation, and proteins, which function is subject to post-translational modifications, metabolites are direct signatures of biochemical activities. They are extremely sensitive to external stimuli in the environment, or internal, from intracellular, physiological, and they're easier to correlate with the phenotype. Genotype indicates what might happen, while metabolomics is in turn indicating what is happening. So the metabolome as a whole defines the final products of gene expression, protein activities in a given moment and condition. And this is one of the reasons that this approach has been adopted widely in clinical diagnostics. Since most metabolites are generated by enzymatic proteins that result from gene expression, and metabolites give organisms their biochemical characteristics, the metabolome links really the genotype with the phenotype. And due to this reason and close connection between the genome and the metabolites, the metabolome is called, or the metabolites are called, the canaries of the genome. One single DNA-based change in a protein coding gene, especially in metabolic enzymes, can potentially lead to a 10,000 amplification in the concentration of a metabolite. And another thing that we have to take into account is that to assemble genes, there are 4 bases that we need. There are 4 nucleotide bases that we need to assemble proteins like Christine, Yuchen, and Ibet talked yesterday. We need 20 amino acids, while the diversity of chemical classes in the metabolites is huge compared to the other numbers. Metabolites can be endogenous, which means they're routinely produced by anabolism or catabolism. These molecules are also called the primary metabolites, and they're essential for growth and development, for example. And some typical metabolites that are endogenous are amino acids, sugars, organic acids, but they can also include xenobiotics that are coming from diet and the environment. Some examples of these xenobiotics compounds are plant phytochemicals, food additives, pesticides, and chemical contaminants. When we have our cultures in the metabolic engineering experiments that we are discussing, these external metabolites are present in the medium. The amplification phenomena that we were describing before for the components of the genes, proteins compared to the metabolites can also be observed when we think about the number of metabolites that are present in an organism. So for microorganisms, that kind of similar to the ones that we'll focus in the talks in the afternoon in the tutorials, for these microorganisms like E. coli, cerevisiae, there are estimates between 4,000 to 16,000 metabolites exist. So this would be the total metabolome in this type of organisms. Individual plants are thought to have between 30,000 to 80,000 metabolites. And if we think about the entire plant kingdom, the estimates are for about 200,000 to 1 million of metabolites. So these numbers should tell us about how large and complex the metabolome is. After going about these basics of metabolomics, I want to move a little bit to the history of metabolomics, or how did it start. So we've been wanted to measure metabolites since approximately 2000 before Christ. So in that time, traditional Chinese medicine was using ants to evaluate the urine of patients. And at the same time, Hindus were also recording that insects and flies were attracted to the urine of some people, and that the urine tasted sweet, and that this fact could later be associated with certain diseases. In the Middle Ages, urine charts like the one in the left were used to correlate smell, taste, and color of urine to diagnose various medical conditions that are metabolic in origin. Now, moving to more recent times, in the 40s, Roger Williams suggested that each person had a metabolic pattern, and he measured it in fluids such as urine and saliva, and he was doing, his studies were on patients with schizophrenia. In the 70s and in the 80s, there was a huge breakthrough related to the GCMS development, and it was possible to measure compounds in urine and tissue extracts using this technique. The term metabolic profile was arrived during this time too, in the 70s, 80s, and a huge new player got to the field in the 80s. So this player was NMR. NMR applications on metabolomics started in the 80s, and we will discuss NMR in depth in the following sessions today. But an important aspect of this is to remember that omics, metabolomics, is a technology-driven discipline, which is constantly evolving, constantly growing, and it's taking advantages of novel developments in instrumentation, techniques, analytical software, and statistical methods. This figure is a little bit outdated, but we can see that metabolomics is trailing behind proteomics and genomics, which are more popular, and metabolomics is considered younger compared to these other two techniques. So yes, we can see, yeah, the red line is the metabolomics papers that have been published up to 2016. And I was checking yesterday night in PubMed, and for proteomics for the year 2020, I could see around 14,000 papers were published, while for metabolomics, it was 9,000. So it's still growing, metabolomics, and there's room for growth. However, despite this growing interest, we're very aware of the challenges that come with diving into metabolomics. So I'm going to talk in this next section of the talk about challenges that are particular to metabolomics. The first challenge that we face is that metabolites have a large chemical diversity. Unlike genomic and proteomic methods, the objective is to measure molecules that have completely disparate physical properties. Some of the examples are shown in this slide. These diverse physical properties make the metabolites much more differentially extractable, and also many of them are unstable. So to date, there is no unique analytical technique that can cover the entire metabolome. Consequently, a combination of different extraction techniques and analytical methods has to be used so we can reach an adequate metabolite coverage. Accordingly, if we want to do comprehensive metabolomics, we usually divide the metabolome into subsets of metabolites. And this is, for example, the approach that we use if we're doing LCMS. And we have to think about what's the column that we want to use depending on the metabolite that we want to study. These divisions are often based on compound polarity, common functional groups, or structural similarities. And the different groups of metabolites usually require specific sample preparation and analytical procedures that we have to optimize beforehand. The second challenge that I'm going to discuss is the massive diversity in the concentration found in metabolites. So in this figure from a paper published in 2016, we can see the metabolite concentrations that were reported in human plasma and urine studies according to the Human Metabolome database. As you can see, the variation goes from picomolar to millimolar, and this large concentration range sometimes to account for this large concentration range, we can use parallel analysis of samples. We can try different dilution series, but many times in clinical studies, you don't have that option. You just have a certain amount of microliters to analyze. So that's a challenging part, the massive range in concentration that you can find in metabolites. And this example is just for humans. So imagine for plants or other types of organisms. And finally, as a third challenge, despite all the advances in technologies and informatics, metabolomics analysis is still a very time-consuming process, and identification of metabolites is a limiting factor. So right now, there are more metabolites than are currently mapped on known biochemical pathways. This is the figure that Christine was showing yesterday. So yes, we're getting a grasp of metabolites in these different pathways, thanks to the advances in technologies. And we're illuminating these pathways that are known with the new analytical technologies and bioinformatics tools, but we're still far from done with the identification of the metabolome because we're aiming to measure everything. We still have many unknowns. There's a metabolic dark matter that we still don't know about. When we run samples either by mass chromatography, mass spectrometry or NMR, there are still many compounds that we cannot assign. So now, so yes, that this metabolic dark matter still exists, and we required a lot of development in mostly bioinformatic tools to help us solve this puzzle. So now enough of the challenges that we have with metabolomics, and it's a good time to move to the how. How do we actually work in metabolomics? So the general workflow very simplified, looks like this. So it's quite similar to what we were seeing yesterday in proteomics. It stops before we get to the proteomics point. Of course, what you're changing in this question, what you're changing in this case or in this workflow are the questions or biological systems that we start with, but the steps in general are similar. We start with the sample, which for our tutorials, it's going to be fungi grown in a shake flask, and we have to isolate or extract the metabolites from it. We get these extracts and then we prep them for chemical analysis. We are going to spend a lot of time today on this chemical analysis side. We're going to look into mass spectrometry and NMR, and also on data analysis today and tomorrow in Thursday session. So today I want to talk a little bit more about the first steps that are also crucial so we design a good metabolomics experiment. So as we were mentioning before, metabolomics are a great readout of what is happening in these samples, of what is happening in this shake flask, in this bioreactor, or in the different tissues that we have here. But the changes are drastic due to external or internal stimuli. So changes to nutrients, stress, disease happen long before in the metabolome compared to the transcriptome or proteome. Therefore, if we want to get metabolomic samples that show us a snapshot of what's happening at the moment, we have to quench the metabolism. So quenching just means that it's a rapid inactivation of all the biochemical and enzymatic activities in the cell or in the organism. As I mentioned, we want to get an accurate picture of what's happening at the moment with this tissue or with this sample. And there are different ways in which we can do this quenching. The most common involves using liquid nitrogen and cold solvents. As you can see, I lost my pointer. As you can see in this table by this study in 2014. So there are different methods that you can use. Usually they involve liquid nitrogen or solvents or changing the pH. And there are many articles comparing quenching methods in different biological systems. So it's always a good idea to start checking those before you perform your experiment. Once the samples are quenched and safely stored, usually at -20 or -80, we proceed to the sample extraction. So extraction is a critical step in metabolomics. And as with quenching, you're going to find lots of options regarding solvents, solvent ratios, temperature, and use of beads, and even the materials of the beads can be different depending on the biological system that you have. These again, yes, this depends on the tissue or the sample that you have, but again, you'll find a lot of protocols that you want to check before doing your experiments of metabolomics, because you cannot think that one is going to work for the others. So the method that we use for the samples that we'll be studying, and for most of the samples here, it's called MPLEX and was described in yesterday's sessions. So yesterday, the focus was on proteins, on this protein disk, and we heard everything about digestion and the different ways that you can prep this protein disk that you get between the phases. But now today we're going to focus on what happens with the upper and lower phases and how can we analyze the metabolites that are present on those phases. We have lots of options of instruments for analysis. So these are some examples of instruments that we routinely use in metabolomics for either untargeted, targeted approaches to get relative levels or absolute quantification of metabolites, and if we're doing 13C metabolic flux analysis. You'll notice a couple of acronyms in common, and those are mass spectrometry, MS, and NMR. Today, we're going to talk in detail about these two analytical platforms. We'll have the talks in the morning and the tutorials in the afternoon dedicated to mass spectrometry and NMR. I'm going to go briefly about a comparison between the two, but you'll hear more about it this morning. So in this table, you can you can see that sensitivity is one of the main points of comparison between the two, with mass spectrometry, especially LC-MS and the data independent acquisition being the best approach relative to sensitivity. However, there are other important points if we want to do a fair comparison. So in NMR, the reproducibility is very good, while in MS, it's not as good. So in this case, yes, NMR is better in terms of reproducibility. Mass spec, in the other hand, is a destructive technique. So the samples are ionized, they're vaporized, while in NMR, they're not. Samples can be recovered and measured again and again. So not destructive NMR, while we have a destructive technique in MS. The sample preparation for MS in general is more involved compared to NMR, and steps like derivatizations are not required in the case of NMR. In MS, sample needed is usually in the low microliters or milligrams because of the sensitivity, while in NMR, you usually need more volume or biomass because, however, there are new developments in this field that we'll hear about this afternoon, this morning, sorry. And finally, in this very brief comparison, MS has excellent databases while NMR libraries are still in development. So as you can see, each technique has its pros and cons. The list is way longer than this, but in general, they're complementary. It's known that there's no universal analytical technique for measuring the two. So combining the power of both is the ideal situation in a metabolomic scenario. So, okay, moving to the type of experiments that you can do when you're doing metabolomics. So with both platforms, MS and NMR, we can do targeted or untargeted metabolomics approaches. They're both possible. We use targeted metabolomics when we want to answer questions about the levels of certain metabolites that are present in a sample. We don't want to measure all. It's a very specific question. In this case, we need to have standards of the metabolites. If we're doing MS, this diagram is about mass spectrometry, so I will talk about the mass spectrometry angle. But if we're doing MS, we need to acquire the mass spectra of the metabolite. We need to know its retention time, for example. And if we're doing LC-MS, we want to select what are the transitions that we're going to use for the quantifications. Because we're going to be focusing on a smaller set of metabolites, we want to optimize the method. We can usually make the runs shorter, and we will have to generate standard curves to the quantification. We can study using these targeted metabolomics tissue lysate, cells, blood, and other biofluids. And then once we have our method developed, we have the standards, the standard curve. We can acquire the LC-MS or the GC-MS of the metabolite extracts. Then we can compare the sample groups that we have based on intensity, peak areas, and then using the calibration curve that we generated, we can quantify the different metabolites that are present in these samples. We use targeted approaches in metabolomics with these very specific questions. So just recapping, remember that this is the set that we're studying in the tutorials, and this is Aspergillus pseudoterus strains. And if we want to measure the carbon, the glucose, which was the carbon source consumed over a course of 8 days, or the production of the 3-hydroxypropionic acid, this is the approach that we use. And also, as we mentioned in day one, to evaluate microbial bioproduction, we can measure titer, yield, or rate. And in all of these cases, for these measurements, we need targeted approaches. And finally, for in a paper that was using a similar strains to this, we use targeted approaches to measure all of the different metabolites that were also generated in the medium by these fungi. That is the approach that we follow when we're doing targeted metabolomics. What happens if the question is broader? If we want to get the global profile of the samples, if we want to measure all the metabolites that are present, in this case, we're going to go to untargeted metabolomics. We don't need to run standards if we're following an untargeted approach. However, in this case, so that's why we're not showing here the standards. We just go straight to, of course, we have to still do the quenching, still do the extraction, but we measure the sample. And now the big effort is going to be in the data processing and analysis. Validation of the identifications is also crucial. And if we're using LCMS, we usually acquire in MSMS mode, and we have to validate these identifications using a library that we will discuss some examples in a second. So when we use this approach, we get a global metabolic profile of the biological samples that again can be tissue lysates, cells, or biofluids. So when we do untargeted metabolomics, we get relative levels of the metabolites detected, metabolites that in this example are part of the modified pathways. So yes, again, we get relative levels, big areas, but we get a lot more than that. We get big areas of hundreds of identified metabolites, and we also get a lot of unknowns. So to get to this, and this is an example of yes, Aspergillus pseudoterus, and we collected all of the features that are in the green box where metabolites identified are unknown in these metabolite extracts from Aspergillus pseudoterus. To get to this metabolite identification, which we highlighted as one of the challenges in metabolomics, we need to use bioinformatic tools. We cannot do this manually. So I'll discuss briefly what are the steps in word processing metabolomics data. So Will Q and in the tutorials we will cover this part, but very simplified, the two steps are these. First, we need to process the raw metabolomics data. In this step, we find features and we group them between samples, and we have these tools shown here. Some of these tools are proprietary, others are open source, and they support different kind of data in mass spectrometry. This list is not comprehensive. There are many that are still missing, and I'm not including here the available tools in NMR. Processing requires steps like the convolution that we're going to see in detail today. So up until this first step, we're getting until here, where we're processing our raw metabolomics data, find common features, group them, and we get to this step. But in the second step, which is analyzing the software results obtained in the previous steps, we have to remember that this software usually, the previous software usually gives us a list of MC values, profiling in MS1. These MCs have to be searched in databases like the ones shown here to find suggested compounds that match these mass. And we have to validate the potential compounds comparing to MS2 data and other parameters like retention time, CCAs, if we're using specific analytical techniques. So yes, some of the software available for this step is shown here. Each has its pros and cons, and you can find even articles describing what are the pros and cons of each, and we'll discuss some of them in other sessions today. They use different fragmentation methods, precursor ion types, and there are thousands of compounds of analytical interest. Medlin, for example, right now is nearing 1 million of molecular standards with MSMS data acquired at multiple energies, but each of them, yes, as I mentioned, has its strengths and weaknesses, so sometimes works better than the other for your specific project. And still, the main goal is addressing that third challenge of unambiguously identify metabolites. So applying the concepts to the school topic. The results from untargeted approaches metabolomics, shown here, and the results from targeted measurements like the 3-HP production in these strains can learn with a clear picture of what is happening inside and outside of the cells. An integration of this data with other data like proteomics can help new rounds in this design, build, test, learn cycle, and at the end lead us to higher TRY metrics, which are these titer rate and yield metrics. With this, this is like the general part of metabolomics, but now I want to spend some time talking about other omics that we do in the lab, new technologies that are going to be implemented. So I'll start with lipidomics that I mentioned briefly on Monday. Lipidomics is an even younger omic than metabolomics. From the figure that I showed before, when they were growing, lipidomics started way later. It's an area with tremendous potential. There are many thousands of lipid species that exist, and their metabolism is linked via numerous pathways and networks. And these networks, just as with metabolites, they can change in response to cellular environment alterations such as exercise and development of a disease. Measuring these alterations and understanding the pathways involved is crucial to understand cellular metabolism. And to lipidomics, we get if we're doing lipidomics specifically, we get the lower phase of the MPlex extraction, and we use mass spectrometry tools in lipidomics. So in LC-MS, a key bottleneck is to process the spectral data as with metabolomics in general, in a high throughput manner, and at the same time providing confident identification and levels of these lipid species. Therefore, the lipidomics team in the lab developed a tool called Liquid, which has a very large library. It has more than 35,000 lipid molecular species. It is customizable. You can add your novel lipids if you have the standard for, and it has the ability to identify separated isomers. It has the ability to identify LC separated isomers as well as identify co-eluting species. And this is how the graphic user interface looks. A new development in this area in the lab that the experts here are implementing is ozone-induced dissociation to locate double bonds in lipids, like the one shown here. This is one of the most significant challenges in contemporary metabolomics, the separation and identification of lipid isomers that differ only in one side of unsaturation or the geometric configuration of the carbon-carbon double bonds. Ion mobility, which is also studied in the lab, presents a fast and scalable toolkit that is orthogonal to LC and can be used to enhance separation. So to address this challenge, ozone-induced dissociation in line with LC, IMS, and high-resolution mass spectrometry will be implemented, and samples are acquired without the ozone, with the ozone, and the diagnostic ions generated by the ozone use combined with the other measurements about CCS and drift time generates a rich data set that carries information about the site of the unsaturation across all major lipid classes. So they're right now working in the modification of the ion mobility instrument to be able to do these measurements. Another technique that we won't cover here today is imaging mass spectrometry. And it's very nice because one of the very first steps when we're doing metabolomics applied to biological tissues or cells is that we homogenize the sample. To do this, we use different, we can use different bits, or we're even in the MPLEX, we're combining everything. So we have all the metabolites from the tissue basically in the same vial. So standard untargeted metabolomic approaches don't allow high resolution spatial localization of metabolites within the samples. So research in heterogeneous tissues like organs, even plants, are complicated because you're just combining everything in the same vial. So matching dysregulated metabolites with specific regions, it's going to be basically impossible if you have variation in your cells. So mass spectrometry imaging is a technique that can help with this issue. Using mass spectrometry, we can do an untargeted investigation into the spatial distribution of molecular species in a variety of samples. Thousands of molecules, such as not only metabolites, but also lipids, peptides, proteins, can be measured in a single experiment without labeling, and it requires careful preparation. And at the end, when you get the spectra, you can use computational software to select a specific MC value, and you can generate heat maps that show you the intensities of the metabolites or of the molecular species present. And identification is done after that based on MS or by intact mass if you're using database. This is I'll go briefly over this because I know I'm running out of time. So this is an example of a recent paper published by Dushan here in the lab, and they use spatial lipidomic experiments to track lipid changes that occur locally at the site of leaf injury. So they were sowing tomato leaflets that were crushed using surgical forceps, and then the leaves were cryosectioned and analyzed. And this was done to understand the potential of linoleic and linolenic acid from individual membrane lipids, from where membrane lipids this linolenic acid could be found, because it's usually found in the injured side of the leaves. So it was found that phosphatidylcholines strongly colocalized with the injured zones of the wounded leaflets, and similar localization was also found for LPE and lysophosphatidic acid. So the high resolution, in this case, high resolution colocalization imaging suggested that linolenic acids are predominantly released from PCs with 16-18 fatty acid composition along the entire leaflet. And finally, just to close this, we'll talk about a little bit about stable isotope assisted metabolomics. In these approaches, when we follow this approach, we use heavy labeled isotope tracers 13C, 15N, 18O to support studies of biochemical mechanisms. By measuring the incorporation of this tracer into the metabolites that are produced downstream, we can identify and discern pathways involved in biochemical processes and even metabolite reaction rates. We have to remember that in metabolite profiling metabolomics, the measured concentration or the levels of the metabolites are the summed abundance of the metabolite if the metabolite is synthesized in more than one pathway, which is usually the case. So the relative contribution of the pathways to the synthesis of that metabolites remains ambiguous. Therefore, the abundance measurements are frequently insufficient if we want to get an exact and accurate pathway assignment and identify the the changes in metabolites and metabolites fate and flux. So for 13 CMFA, other parts of the puzzle are required, like the metabolic map to be able to reconstruct the metabolic model of the organism if we want to be able to reconstruct fluxes in the organism. These are complex experiments. They require elaborate data analysis and are more expensive because we have to use a labeled source and haven't been as routinely used as the other approaches. However, they're the golden standard in metabolic engineering because they are great to characterize non-model hosts. They can help to identify wasteful byproduct pathways, and they can identify pathway bottlenecks, which is great for other rounds of DVTL cycle and higher TRY metrics. So today, you're going to be learning about the fundamentals of mass spectrometry-based metabolomics with Youngmo in the next talk. Then after that, Robert is going to talk about the basics of NMR-based metabolomics. We'll have, I don't think we'll have a break, but after that, we were going to meet with Will Q, who is going to discuss metabolomics data processing pipelines for both MS and NMR approaches. And finally, for the afternoon tutorials, we're going to processing MS and NMR data applied to an Aspergillus pseudoterus dataset. So that is it for my talk this morning. Thank you very much. I'll be checking for your questions in the chat and in the Discord.
Thank you for the nice introduction for metabolomics. And there are a lot of questions in both the Zoom chat window and the Discord. And I see a lot of questions are being answered. Nathalie, can you look at the questions?
I'm looking at them.
Yeah, I have a list of the questions here. I can read for you, Nathalie, if that will help.
Okay.
I made a list of what was already answered as well.
Okay.
So the first one is an answer is, if you're starting a new metabolomic experiment with a known preconceived knowledge of what the metabolites might be involved in your study, how do you decide what metabolites to target in your initial pass? Do you rely on a protein gene experiments to focus your attention, or do you investigate multiple metabolite classes to determine what metabolites are related to your study? Question. It's a big question.
Youngmo, did you read that one? I kind of got lost trying to make the laser pointer disappear. But we usually do that an untargeted metabolomics first. And then based on the ones that are the most abundant, if we want a specific quantification, then we can run the calibration standards and the calibration curves to get the accurate number of them. So it depends if you really need those grams per liter, if you really need that concentration, or if you just need to compare the levels between one and the other. And if you have like GCMS or with a large library, you can definitely compare them enough with an untargeted one without doing the targeted quantification. In the targeted, you need to run the calibration curve. In the untargeted, you just need to run the sample extracts. So it was that one from Discord. I think
yeah, that's the first one on Discord.
Yes. Okay. Yes. I don't have that one open. So I'll check it in detail and I can reply in the directly to the person. I'll just reply there so everyone can see the answer.
I think we still have a minute for more questions, right? Okay. So another one that it's not answered is, does the metabolome constitute the most abundant class of compounds in terms of total moles of metabolites in a cell compared to other classes such as transcriptome or proteome? The broader question is there a way to appreciate the relative abundance of this class of omics compounds? I think you answered that on your presentation, but if you can touch back on that.
Yes. You can optimize your method for specific metabolites, but if you just do untargeted metabolomics, because of the way that they fragment like each metabolite, it's going to be difficult to say there is mostly or of this class based on compared to the others. So yes, no, I don't think that is that possible. unless you have some media that it's just like glucose or something, so you can see, okay, there's only this metabolite present. But yeah, I would have to read it. It's kind of tough to.
Okay.
I'll go to Discord while we have the break after Youngmo's presentation, and I will try to reply to those ones.